In this video, we will discuss about a metabolic enzyme, that is IMPDH2, that helps in cancer cell DNA damage repair. So far in the biology, people have studied that the different compartments of the cells plays a different role. But this paper that is published in the Nature recently has shown that there is a interrelations also occurs. And one of the examples of those interrelations is shown in this paper, where it has shown that this metabolic enzymes is been recruited to the chromatin whenever the cells experience a DNA damage. And they did their experiments on the triple negative breast cancer cells. We will discuss those results in this video and also in the subsequent video one by one. So this is the first figure that is presented in the paper, where they have shown that there is upregulation of this IMPDH2 in the triple negative breast cancer cells. And what are the other things that they have shown in this figure, we will discuss it. So in the panel A, they have checked the markers for this in these triple negative cells and also the other breast cancer cells. And what they have find out is that they have seen the DNA damage is more prominent in the triple negative cells. So in the panel A, what they have checked the DNA damage in different kind of breast cancer cell lines. And from the literatures, they know that triple negative cells are more prone to DNA damage. So this is the triple negative cell line name, that is MDA MB231, and these are the other breast cancer cell lines. And this is the markers for this gamma H2AX is the marker for the DNA damage. And what they have found out is that DNA damage is higher in the case of triple negative cells compared to the other breast cancer cells. And this is in line with their the literature that has shown that DNA this triple negative breast cancer cells are more prone to DNA damage. And these things from the immunofluorescence image, they also did their quantifications, and it also shows the same thing, that triple negative cells has more DNA damage compared to the other breast cancer negative cells. And when they check the aggressiveness of these cancer cell lines, what they have find out is that the aggressiveness of this triple negative cancers is highest, and it goes down for the other cell lines. And once they have identified that DNA damage is more prominently expressed in the triple negative cells, they wanted to find out the reason why this DNA damage is more prominent in those cells. So what they did in this case is that they took they did the mass spectrometric analysis of the cell lines from these triple negative cells and compare it with the other breast cancer cells. And what they have find out is that there is upregulations of the genes that are involved in the chromatin in the case of triple negative cells. And more specifically, what they have find out in the case of triple negative cells, there is upregulations of the genes that are involved in the chromatin, which is expected. And one more thing that they have find out, there is upregulation of the genes that are involved in the nuclear localizations, and this is not expected. And when they compare this results of the triple negative cells with the other cell lines, what they have find out is that with the increase in the aggressiveness of the cancer cell lines, there is a high upregulations of this IMPDH2. That is in the first case, the comparison shows that MDBA to the triple negative cells has around this levels that is shown by the red arrow, has this level of upregulations. But this upregulations increases when they did the estimate of only this nuclear localization cells. So So this indicates that. So this first one is this there's just the volcano plot that shows the comparisons of MDA MB231 and MCF7, and the other figures, that is this panel right hand side figure of the penalty shows the overall comparisons, and it shows that IMPDH2 is highly upregulated when they compare with the other less aggressive cell lines. So in order to validate this point, what they did is they did the fractionation of the cells in two categories. That is, first one that belongs to the cytoplasmic region, and second one belongs to the chromatin region. And the markers that they use for cytoplasmic region is vinocilin, and markers that they use for the chromatin region is this H3. And what they have find out is that in the case of different cell lines, the IMPDH2 is highest when they check it for MDA MB3231 in the case of this cytoplasmic region, and for the when they check this same IMPDH2 in the chromatin regions, they can see that in the chromatin regions also there is some presence of IMPDH2 for this breast cancer cell lines, and which is slightly not expected, as this IMPDH2 is usually found in its usual location is in the nucleus, though it is also expressed for the other cancer cell lines. So this could also means that in the other cancer cell lines there is a DNA damage more prone to it. So in order to validate their hypothesis that the IMPDH2 might be recruited by the cells whenever there is a DNA damage occurs, what they did, they checked the they wanted to check this hypothesis using a model that is shown in the panel F, where they took the MCF7 cells, and in the literature it is mentioned that if we grow these MCF7 cells in the hormone free media, it will convert it into the MCF7 HI cells. This means that the cells will acquire more aggressive characteristics and behave similar to the triple negative cells. So this MCF7 HI cells should also reflect high upregulations, or should also reflects the upregulations of this IMPDH2 in the chromatin region compared to the MCF7. So what they did is they grew the cells MCF7HI, and it has a mixed population. Some of them are high proliferative, some of them are not high proliferative, and they segregate these high proliferative cells using the DAPI and nucleus intensity, and thereafter they did the Western blot, and what they have find out is that in the chromatin region, they are able to see more IMPDH2 compared to the this MCF7 cells that can be seen here. The more proliferative cells has more IMPDH2 signals compared to this MCF7H. Let me reconfirm it. Yeah, so this is the cytoplasmic region. Here we have seen that vincerin is there and IMPDH2 is there in both the cases. And for the chromatin region, they see that high proliferative cells has more of this chromatin compared to the non-proliferative cells of this MCF7. And these things they have quantified. And thereafter they also checked these DNA damage signals using this marker gamma H2AX, that is the same marker that is used in the panel A, and what they have find out is that DNA damage is more in the MCF7HI compared to its parent, that is MCF7. And after that they check around 114 samples, and what they have find out is that the cancer cells which are more aggressive in the patients has more IMP DH2, that is patients that have this triple negative breast cancers symptoms has more IMP DH2 compared to the non-triple negative cells, and this is statistically significant. And the immunostaining image is also shown that the triple negative cells, that is HR positive triple negative cells compared to the HR positive cells has more of this IMP DH2 signals. So their hypothesis that IMP DH2 is upregulated whenever the DNA damage occurs in the cancer cells, especially in they checked this hypothesis in the triple negative cells. It seems to be correct till this point of time. So I hope you understood the results that are presented in this figure. And if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.